You're welcome to BTG 202 class. I'll be looking at historical development of biotechnology. In this lesson, we will learn the following about the developmental history of biotechnology, meaning of biotechnology, branches of biotechnology, brief history of biotechnology, and developmental stages of biotechnology. Meaning of biotechnology. Biotechnology is a combination of biology and technology. So when you combine it together, we are saying that it is the use of microorganisms by using a technology to produce a finishing product. Biotechnology is the use of organisms to make a product or a process, etc. Let us look at the branches of biotechnology. Number one branch of biotechnology that we have here is animal biotechnology. These deal with the development of transgenic animals for the purpose of producing milk and also meat that will actually be resistant to diseases. It also deals with the in vitro fertilization of eggs and transfer of embryo in animals, which including man, for further development. We also have one branch of biotechnology which is referred to plant biotechnology. This plant biotechnology we normally refer to them as green biotechnology, and it deals with the combination of tissue culture and genetic engineering. It also deals with the development of transgenic plants with resistance to biotic and abiotic stress, development of haploid, embryo rescue, clonal multiplication, and cryopreservation. It is very important to say that the animal biotechnology and plant biotechnology are agricultural biotechnology. I will normally refer to them as green. Another branches of biotechnology is medical biotechnology, which we always refer to as red biotechnology. It deals with the diagnosis of various diseases, large-scale production of various drugs, vaccines, and hormones for the treatment of various diseases. Another branch of biotechnology is industrial biotechnology, which we normally refer to as white biotechnology. This kind of biotechnology is used and applied in industry and in processes. Ordinarily, we use microorganisms in the industry to produce different kinds of the product. It also deals with the commercial production of very useful organic substances, such as chemical. We have citric acid and so forth and so on. We also have environmental biotechnology, which we call blue biotechnology. It deals with the detoxification of waste and industrial effluent, treatment of sewage water, and control of plant disease and insect through the use of biological agents. Ordinarily, bioremediation actually comes in. And when we're talking about the bioremediation, we're talking about using microorganisms to remove a contaminated waste from the environment. And the last type of biotechnology is what we call bioinformatics. This is the combination of computer and biotechnology, and it helps in finding the analysis of data related to biotechnology. It plays an important and a vital role in areas like functional genomics, structural genomics, and protein. Let's look at the brief history of biotechnology. Now, people were using biotechnology techniques thousands of years ago, but they did not really know that they are even using biotechnology. The name biotechnology was given by Hungarian engineer Károly Ereky in 1919. Biotechnology arose from the field of zymotechnology or zymogen, which we normally refer to produce a new product. By World War I, zymotechnology has expanded to tackle large industrial issues. Therefore, the ancient Egyptians made wine using fermentation techniques and conversion of even yogurt to milk. People also use selective breeding and all this technology. Now, let's look at the developmental stages of biotechnology. The developmental stage of biotechnology is subdivided to three. One is ancient biotechnology. This is pre-1800. We normally refer to it, we call it earlier history as related to food and shelter, which include domestication of animals. This stage is termed a discovery or developmental stages. Another developmental stages of biotechnology is classical biotechnology. This one takes place between 1800 to 1900. This phase was timed evolution and development. Various observations started pouring in with scientific evidence, and they build on ancient biotechnology. The last one is modern biotechnology, which normally happened between 1977 up to today. This culminated with the birth of genetic engineering, and there were two key events. And when we are talking about genetic engineering, we are talking about manipulation of DNA. Now, the two key events that happened, one was the 1953 discovery of the structure of the DNA, and the other one was the 1973 discovery of the recombinant technology. In our next class, we will look at we'll build on this modern biotechnology. Thank you for listening.